The complement cascade is part of human immunity system to fight against pathogens using plasma components. There are three pathway complements can be activated on pathogen surfaces, namely the classical pathway as the first pathway to discover, alternative pathway as the second pathway to discover, and lastly the newest discovered mannose-binding lectin pathway. The complement cascade is part of human innate and acquired immunity. First, the activated complement component is able to bind to pathogen and activate opsonization. Second, fragments generated by complement act as chemoattractants to recruit phagocytes. Third, the terminal components of complement is able to kill pathogen by creating pores in membrane. Let us go through the classical pathway of the complement cascade. There are two ways to activate the classical pathway. C1q of the C1 complex can bind directly to the pathogen surfaces, or C1q of the C1 complex can bind to the antibody-antigen complexes of the pathogen. The binding causes a conformation change in C1 complex and activates C1r and C1s. C1 will initiate a cascade of reactions involving the complement protein C2 to C9 as follows. Activated C1s is a serine protease which cleaves C4 into C4a and C4b. C4a is a small fragment with the role of weak pro-inflammatory properties, while the larger C4b is able to bind covalently to pathogen surface. Upon binding of C4b to pathogen surface, C2 is able to bind the attached C4b and is cleaved by activated C1s serine proteases into C2a and C2b. C2a is a small fragment when further cleaved into C2 kinase will cause extensive swelling. C4b will combine with the larger fragment C2b to form C3 convertase. The C3 convertase is able to bind C3 and cleave C3 into C3a and C3b. C3a initiates a local inflammatory response, while C3b combines with C4b, C2b to form a C5 convertase. The C5 convertase is able to bind C5 and cleave C5 into C5a and C5b. C5a is a peptide mediator for inflammation, and C5b is able to initiate the late events of complement activation. C5b binding to the pathogen surfaces will cause C6, C7, C8 and C9 to form a membrane-attack complex that creates a pore on the cell membrane of pathogens that disrupts the ionic and osmotic balance and kill the pathogen. Here we have come to the end of the classical pathway of the complement cascade. Let's move on to the second pathway known as the alternative pathway of the complement cascade. Factor B, D, and P are involved in the alternative pathway. The alternative pathway is generated through spontaneous hydrolysis of C3 to C3b. C3b will remain bound to the pathogen surface while C3a is already dissociated from C3b and the pathogen surface. Factor B will bind to C3b cleaved by factor D into BB and BA. BB remains bound to C3b while BA will dissociate from the complex. Factor B is able to bind C3b BB complex to form a stabilized C3 convertase. The C3 convertase then fits into the C3 convertase pathway downstream of the classical pathway. There are few components that can inhibit the alternative pathway. The complement receptor CR1, a membrane-attached protein known as decay accelerating factor DAF or CD55, as well as factor H, is able to compete with factor B for binding to C3b on the cell surface. By disrupting C3b and factor BB association, this will prevent the formation of C3 convertase. In addition, factor I, which is a plasma protease, in conjunction with C3b binding cofactors such as CR1, membrane cofactor of proteolysis MCP or CD46, and factor H, is able to cleave C3b into its inactive IC3b derivatives to prevent C3 convertase formation. Finally, let's go through the last pathway, which is the third pathway of the complement cascade, known as the mannose-binding lectin pathway. The mannose-binding lectin pathway is homologous to the classical pathway. The mannose-binding lectin binds to the mannose on the surface of the pathogen and activates MBL-associated proteases MASP1 and MASP2 to trigger the cleavage of C4 and C2 complex in the same manner as the C1 complex of the classical pathway for the formation of C3 convertase. The formation of C3 convertase in this mannose-binding lectin pathway will then feed into the downstream of the classical pathway. To summarize, these are the three pathways of the complement cascade.